Hello and welcome to Learning Made Easy. Today we'll be discussing about the determination of antibody structure. Immunoglobulin, also called antibodies, are the antigen-binding glycoproteins belong to the immunoglobulin superfamily synthesized exclusively by B cells. Deduction of antibody structure. Antibodies were first identified as serum immunoglobulins in experiments performed by Arne Tiselius, Perce Peterson, Michael Heidelberger, and Elvin Cabot in 1939. Before their experiments, the chemical nature of antibodies in blood serum was unknown. The process of immunoprecipitation, where antibodies react with a multivalent protein antigen to form a multimolecular cross-linked complex that falls out of solution, was used in the experiments. The experiments involved immunizing rabbits with ovalbumin, a protein found in egg whites, and then dividing the obtained anti-ovalbumin antiserum into two aliquots. The first aliquot was subjected to electrophoresis, which showed four major protein subpopulations: albumin, alpha globulin, beta globulin, and gamma globulin. The second serum aliquot was mixed with ovalbumin, and the antibodies in the serum bound to it, forming a precipitate that was removed by centrifugation. The results showed that most of the anti-ovalbumin antibodies could be classified as gamma globulins, and that antibodies of other classes are found in the gamma and beta globulin peaks. The class of serum protein into which antibodies fall was known, but appearance was unknown. Ultracentrifugation experiments estimated the molecular weight of IgG antibodies to be around 150 kDa. First experiment Porter used papain to digest IgG, which produced 3 fragments. The third fragment was named fragment crystallizable, FC, 50 kDa, and demonstrated to not participate in binding reactions. The other 2 fragments were named fragment antigen binding, FAB, each of 45 kDa, and demonstrated to retain the antigen binding capacity of the original antibody. Gerald Edelman used pepsin to produce a single fragment, named FAB2, 100 kDa, that had 2 antigen binding sites. Alfred Nizonoff used beta-mercaptoethanol. Reduction and alkylation of IgG showed that it consisted of 2 heavy chains, each of 50 kDa, and 2 light chains, each of 22 kDa. Experiments showed that antigen binding site, FAB, was made up of both H and L chains, and the Fc part of the molecule was made up of H chains only. Sir Rodney Porter and Gerald Edelman were awarded the Nobel Prize in Physiology and Medicine for their discovery of the structure of immunoglobulins. The overall structure of the antibody molecule consists of 3 compact regions and a flexible hinge region, with the binding sites at the amino termini of the heavy and light chain pairs. Antibody structure. These are Y-shaped molecules with 2 identical antigen binding sites. These have 4 polypeptide chains dotted as a heterodimer and consists of 2 identical light, L chains, about 220 A, 25 kDa, and 2 identical heavy, H, chain, about 400 A, 50 kDa. 2 heavy chains are linked by disulfide bond. Heavy chains and light chains are also linked by disulfide bond. Both light and heavy chains contain a series of repeating structural units, each about 110 AA residues in length, that fold independently in a globular motif that is called an Ig domain. An Ig domain contains 2 pleated sheets, each composed of 3 to 5 antiparallel beta strands, held together by disulfide bridge. Both heavy and light chains consist of amino terminal variable region V and carboxy terminal constant region C. H chain V region has 1 Ig domain and C region has 3 or 4 Ig domain. L chain V and C region both has 1 Ig domain. V region of H and L chain, VH and VL, form antigen binding site. Antibody light chains have two major classes, kappa and lambda. The amino terminal half of light chain is highly variable, referred to as the variable VL region. The carboxyl terminal half of light chain is less variable, referred to as the constant CL region. In humans, 60% of light chains are kappa and 40% are lambda. In mice, only 5% are lambda. All light chains have a molecular weight of 22 keta. V region of L chain has three small hypervariable regions, which were renamed as complementarity determining regions CDRS, CDR1, CDR2, and the most variable CDR3, due to their interaction with bound antigen. The remaining part of V region are known as framework regions, which are relatively constant. There are five major classes of antibody H chains: mu, IgM, delta, IgD, gamma, IgG, epsilon, IgE, and alpha, IgA. Each H chain C region is referred to as an isotype. Isotype of the H chain C region determines its class. The length and molecular weight of the H chains vary according to class, being either 330 AA, 55 to 75 keta for gamma, delta, and alpha chains, or 440 AA and 20% heavier for mu and epsilon chains. The alpha and gamma heavy chains have been further subclassified into subisotypes, leading to the formation of subclasses such as IgA1 and IgA2 for alpha, and IgG1, IgG2, IgG3, and IgG4 for gamma. In mice, the gamma chain subisotypes have been named gamma1, gamma2 alpha, gamma2 beta, and gamma3, and correspond to the subclasses IgG1, IgG2A, IgG2B, and IgG3, respectively. The number and precise positions of disulfide bonds between heavy chains of antibodies vary among classes and subclasses. Antibodies and antibody fragments can serve as antigens. Antibodies from one animal species can be injected into another species, in which the antibody or fragment will serve as an antigen. Antigenic determinants are regions of an antigen that make contact with the antigen combining region on an antibody. The different types of antibodies that recognize immunoglobulin determinants are: anti-isotype antibodies are directed against antigenic determinants present on the constant region of one particular heavy or light chain class of antibody. Allotypes are alternative allelic forms of the same isotype of heavy or light chain genes. Anti-allotype antibodies are generated by immunizing an individual with antibodies derived from another animal of the same species with a different form of the particular immunoglobulin gene. Anti-idiotypic antibodies are directed against the antigen binding site of a particular antibody and can be generated by immunizing an animal with a purified monoclonal antibody. The hinge region is a unique peptide sequence found between the CH1 and CH2 domains of alpha, delta, and gamma heavy chains of IgG, IgD, and IgA antibodies. It is rich in proline residues, making it flexible and enabling the two antigen binding arms of the antibodies to adopt a wide variety of angles. The extended nature of the hinge region makes it vulnerable to protease cleavage. The hinge region also contains cysteine residues, which participate in heavy chain dimerization. The number of interchain disulfide bonds in the hinge region varies among different heavy chain classes and subclasses of antibodies and species. Heavy chains of IgE do not have a hinge region and may inter heavy chain disulfide bonds between the CH1 and CH3 domains. In IgM, disulfide bonds are formed between the pairs of heavy chains at the level of CH3 and CH4. Mu and epsilon chains do not have a hinge region, but have an additional immunoglobulin domain that retains some hinge-like qualities. the 2CH2 domains of alpha, beta, and gamma chains, and the 2CH3 domains of mu and epsilon chains are separated from their partner heavy chain domains by oligosaccharide chains that make the domains more accessible to the aqueous environment and help in binding to complement components. Different classes of antibodies can be expressed as either membrane-bound or secreted immunoglobulin. Secreted antibodies have a hydrophilic amino acid sequence at the carboxyl terminus, while membrane-bound immunoglobulin has three regions: a hydrophilic spacer, (26A), hydrophobic transmembrane segment, (25A.A), and a cytoplasmic tail, (3AA). Immature pre-B cells express only membrane IgM, and mature B cells can switch to different isotypes depending on the cytokines and antigen present. IgG is first antibody formed on antigen encounter. Thanks for watching. If you liked the video, hit the like button and don't forget to subscribe to our channel.